any questions? First, any question? Any question? Is it clear? Anything you want to ask? Question? Yeah.
If you get your tube side or your fallopian tubes, why do you
We're not. Let's
Oh.
let's
Oh,
talk
this
about
question?
this one. We'll talk about this one. You're talking about the practice finals.
Just a question.
It's a question in the practice finals, right?
Oh,
The tubes being tied.
I don't think so.
Yes, it is in the practice
Oh,
final.
okay.
Yeah. So, let's talk about this one first. Okay. Any question on? I meant any question about the announcement about when you're gonna have your final exam. When is it your final exam? Yeah.
Wednesday, 9
right?
a.m.
And it's the same time. You're given 2 hours. Lab time. That's right. During your lab time. Okay. And then what are you supposed to bring for your finals?
2 scan print sheets
Scan.
and some
Yeah.
pencils.
Yeah. Okay. Wash your hands. All that stuff. All. If there's anything, please let me know. I'm still gonna have to make you come in here. If I have to stay until 7 at night, I will stay at 7. You have to come and take your lab exam. Okay. If you want me to come to your house, I can. So, just one more day for you guys. So today and then Wednesday. So we talked about the blood vessels. I think here, I told you it's very important for you to differentiate between artery, capillary, and vein, right? So remember, veins have the valves, and then the arteries have the elastic fibers, and they have thicker muscles. By the way, between the three, arteries are called resistance, right? They're the ones that can constrict and dilate, and that means they're the ones that can influence the resistance. Do you understand what resistance means, right? The pressure you need to overcome. When there's resistance, you can't move. So which of the three can do that? Is the artery, because it can constrict and dilate. So when you see the word vasoconstriction, vasodilation, you're always talking about arteries. You don't even have to think 2, 3, 4 times. It's arteries immediately, right? And then because of these elastic fibers, aneurysm is only an artery problem, never vein problem. In the veins are called capacitance vessels because they have low pressure. You see the muscle is very thin. They can't really push the blood. And so thrombosis is very common in veins only, okay? Thrombosis, embolism, and then capillaries are only made up of epithelium. Actually, just simple squamous epithelium, okay? So we talked about the normal, typical flow of blood. Don't worry about the 3 types of capillaries. What you must know is that exchange, entering, everything, any kind of movement through blood vessels only can happen in your capillaries, not veins, not arteries, okay? Then you must know the typical, so here, table 21.1 is a nice summary of your different blood vessels. You must be able to compute for net filtration pressure, in case that's asked. You already know this one. The key is to know what does hydrostatic pressure mean and oncotic pressure. If you know what those two mean, and you say blood hydrostatic pressure, blood oncotic pressure, you're talking about the pressure inside the blood vessels, right? When you say tissue or interstitial fluid, it's outside. If you know that, you should be good. Okay? Edema means fluid accumulates in the tissues, right, outside of the blood vessels. And this is usually because of decreased oncotic pressure right here in your blood vessels. So what I want you to know is right here, you see the BCOP 26 right here in the arterial end, and in the venous end, it doesn't change. This is your albumin, right? Albumin doesn't leave your capillaries. So the oncotic pressure anywhere should be the same. You know how you move blood in your veins? Muscular contraction, respiratory movement, and then increase abdominal pressure. So blood pressure, hypertension, hypotension. Why is, of the two, which one is more dangerous? Hypotension. Hypotension. Because it's immediate. The symptoms are immediate. If you don't regulate or solve that, or treat that right away, there will be no blood that will go maybe to the brain or to vital organs, and that patient will die, right? Whereas people who are hypertensive, they're still getting blood. It's just at a higher pressure. Then we look at the control, the regulation of your blood flow is by your nervous system and your endocrine system. You all know that. Okay? Baroreceptors, chemoreceptors. Those receptors, whether it's baro or chemoreceptor, when they get stimulated, they will report to the brain, and the brain responds by controlling both pressure and stroke volume and heart rate. Both of them, right, will help. Then, like I said, the blood vessels, if you don't know where the axillary arteries or the femoral arteries at this point, you're in big trouble. But I doubt it. I know you know your anatomy, so you should be good, okay? Okay, the normal blood flow is from the heart to the artery, capillary, vein, back to the heart, right? Then you are familiar with the one special one called portal circulation. You know that artery, capillary, vein, and then another set of capillaries, right? And then another vein back to the heart, right? That's the portal circulation. It's called portal vein. Okay. So just look at this one and familiarize yourselves with these terms. And like I said, I think the only one that maybe you don't know anatomically is the carotid, which everyone knows right here. And then jugular also here. That's the only one I think has no bone counterpart or regional counterpart. So maybe it will help you to remember. Okay. Then we go now to chapter. So the rest of this chapter is just looking at the different vessels. So I'm just gonna jump to the lymphatic system. Go. Okay, so for lymphatic system, you must know the functions. Don't forget, lymphatic vessels are also vessels, but they are not blood vessels. They're different. So, but you call them lymphatic capillaries, lymphatic veins, right? The reason is anatomically, they share the same features, but they're not part of your cardiovascular system. They're different. If they're part of it, then they would be in the same chapter. This is different, okay? So be familiar with the term lymph, right? Then edema, why do you develop edema? So the function of the lymphatic system, where is it? Right here. The lymphatic system, it drains excess fluid. Remember, it just has to return. And then transport dietary lipids because your blood cannot absorb pure fat right away, so you have to convert it to lipoprotein, which happens in your lymphatics, and then immune system. But because the lymphatic capillaries are wider, leakier than your blood capillaries, it's easier for germs and all toxic substances to enter this circulation. So that's why we have the other function of the lymphatic system, which is called immunity. See how it's wider, easier to enter than your typical capillaries. So please remember the lymphatic organs, the main ones, your lymph nodes, your spleen, right? The lymph nodes filter lymph, the spleen filters blood. Both of them have immune cells, meaning your white blood cells, plus the typical or the special lymphocytes, the B cells and the T cells. The B cells go to school or graduate from the bone marrow. The T cells graduate from the thymus. So then, this one, I know I want you to remember MHC proteins. I'm big on that. MHC1, MHC2, CD4, CD8. And then your primary, secondary, first line, second line, and third line of defense. Okay. So first line and second line are part of what you call innate immunity. Innate means you're born with it because they do their job description is the same, whether they're cleaning up a virus or a bacteria or just plain waste material. It's the same, phagocytosis or something like that. Okay, so just remember that it's your third line of defense. That's the specific one, and they're also called adaptive immunity because they adapt. For the second line of defense, you must remember your natural killer cells, which are lymphocytes. So you have B cells, T cells, and then natural killer cells, which are also lymphocytes. But the natural killer cells are part of your second line of defense. And just take a look at, here, summary of innate defenses, table 22.1. Just read it. This first part, the first line, I'm sure you're familiar with it. The second line, just take a look at it, just so you review. Hopefully you remember. Opsonization, antigen presentation. What do those mean? And then in this chapter, you'll see the word agglutination. Complement fixation. So with adaptive immunity, this is your third line, adaptive or specific immunity, and there's two types. The B cells provide humoral immunity, which is immunity in your extracellular space, outside, in the plasma, or it's just outside the cells. The T cells provide what you call cell-mediated immunity, which can help you once the germs have entered your cells, so they can kill the cell. Okay? So the third line of defense is very effective because they are the only ones that's capable of memory. They remember. And it's tailored. The way they respond is tailored to the type of infection that you are fighting. So don't forget these MHC proteins, okay? MHC1, MHC2. And then for cell-mediated immunity or cytotoxic immunity, please know the difference between helper T cell, which is your CD4, and your cytotoxic T cell, which is your CD8. Remember how I told you to remember because the number 4 looks like a letter H. And then the cytotoxic, the 2 O's looks like number 8. So what's the difference between the two? which one is more important. Okay? Remember, the cytotoxic is deadly, but it's useless unless it's called or given the go signal by the helper. So the one that really is more important is the helper. Then the different antibodies. Just be familiar with what does IgG, it says newborns, right here, most abundant, etc., etc., right here. IgA, A for asthma, I don't know, that's what helps me, like, the lining, it's always the IgA, right? Diarrhea, and then IgM is blood typing, IgE is allergy and parasites. Whatever will help you. And this one, table 22.4. Naturally acquired active, naturally acquired passive, and then artificially acquired active, artificially acquired passive. Okay? Next we go to chapter 20, the respiratory system. You did a nice poster on this one, so I hope that you really read this chapter. It's a very short chapter, very easy. Everything. Say to your brain. So, for the respirators, always know the function. Know ventilation, what is ventilation, what's external respiration, internal respiration, what does inhalation mean, and then exhalation. The structures up to the larynx. The larynx is the beginning of the lower respiratory. Right? So when you say you have a URI, it only involves the nose up to the larynx. That's it. So when you have a lower respiratory infection, that's when they become specific. They say you have laryngitis, bronchitis. That's a lower respiratory infection. So be familiar with the anatomy. So the nose, of course, you know why you have the nose. Then you have the nasal concha there to create turbulence. The nose is for olfaction as well as for normalizing the air that's entering, make it adjust it to the normal body temperature and humidity. Then you have the pharynx, which is a shared organ between digestive and respiratory system. So be familiar with the word nasopharynx, oropharynx. The larynx, I don't know why students always, they don't realize that the larynx is made up of these structures. The epiglottis is in the larynx. It's what makes up the larynx. Then you have the thyroid cartilage, and then you have the cricoid cartilage. Those are the three big cartilages that make up the larynx. This one right here, these structures right here, this is now trachea over here, once they're broken. So be familiar with the big cartilages. What is the function of the cartilage? Why do you have cartilage there? To keep your airways open, right? And by the way, the epiglottis is elastic cartilage, but the others are hyaline cartilage. The trachea is C-shaped, and posteriorly, the trachealis muscle completes the ring of the trachea. And then you have two bronchus. Pulmonary infection affects most commonly the right, because the right bronchus is wider, the opening, and it's more direct to the lungs. Then the bronchus branches into smaller and smaller until you get to the terminal bronchiole. Know the lining, pseudostratified ciliated columnar epithelium, then it goes down to cuboidal, then it goes down to squamous in the alveolus. Right? Know that there's hyaline cartilage in the structures higher up, but then the hyaline cartilage stops at the terminal bronchioles, where you only have smooth muscle there. Not terminal, the lower, the smaller bronchioles. Then you go to the lungs. The lungs is made up of simple squamous epithelium. There's two types of simple squamous epithelium cells here. One is called alveolar type 1 for respiratory exchange, and the other one is called type 2 that makes your surfactant, which lowers the surface tension. In the pleura, you want high surface tension. In the alveolus, you want always low surface tension. Then in the alveolus, you have lots of elastic fibers and of course capillaries. Here, there's what you call the ventilation perfusion coupling. What that means is areas of the lungs that have poor ventilation, there will be no perfusion. The purpose of blood that goes there is to pick up oxygen. So any area of the lung that has no oxygen, there will be a shut. Your arterioles will constrict to just redirect the blood somewhere else where there's air, because it's useless to go to a part that has low ventilation. And this is why stroke is very common in people who smoke, or any part. Can you imagine if there is, let's say there's a big chunk here that's been covered by tar, so there will be vasoconstriction there. That's a reflex, you can't control it. There'll be constriction of the arteriole so that the blood will be rerouted. So then that means instead of blood being nice and smooth, spread equally, all of a sudden it will be like crowded in one area, and that's very high chance for coagulation. They start crowding. So that's why it's just not good to smoke, or even with people who have pneumonia or any kind of infection. Sometimes, now you're getting closer, some of you are already practicing. Some people are saying, I had pneumonia, why ended up with a stroke, something like that. Now I hope you understand, there's lots of blood that goes there. And so in pneumonia, that part of the lungs is no longer working. What happens to the blood there? They get rerouted. So now they get very crowded in that area. Okay? It's just it will make sense once you understand what's going on. Okay, ventilation. So you must know ventilation means breathing, then external respiration means from the lungs to the blood, and then when the blood delivers it to the cells, so the cells will use the oxygen, that's called internal respiration. Oh, this is a nice summary of the structures, table 23.1. And then Boyle's law, how do you breathe? It all starts with the chemoreceptors in the medulla. When they detect high levels of CO2, they stimulate your diaphragm through the phrenic nerve and the intercostal muscles to contract, increasing the volume. When you increase the alveolar volume, the pressure goes down, so air comes. Boyle's law, inverse relationship of volume versus pressure. Inhalation is active, exhalation is passive. Exhalation happens because of the recoil of the elastic fibers and the abdominal contents trying to regain their space. Spirometry only measures the lung function, the health of your lungs, okay? So tidal volume is the normal amount of air you breathe in and out. You already know this one. The inspiratory, the reserve, the expiratory reserve, and the residual volume is the amount of air that stays there, that you cannot breathe out, right? To keep the lungs open. So take a look at this one in case there's questions on that. There will be no computation. Usually we ask students to compute for the spirogram, which I think I asked you to compute in, oh no, you didn't have a test for this one. Then once this is what I mean, see the alveolus, the blood goes there to pick up the oxygen. And you must know how is oxygen transported. It's mainly inside red blood cells. The book says 98.5, right? Bound to hemoglobin, the iron part. And then 1.5% is the amount of oxygen that's transported right here in the plasma. So the red blood cells have the majority of its job is to deliver, right? So that's why, what's that? Carbon monoxide poisoning is very deadly because you see, it's compromising the 98.5% capacity of your red blood cells to transport oxygen. So what we do is you give them 1, 2, 3, 400% oxygen to increase the plasma oxygen concentration. It will deliver the oxygen still. This was just a more efficient way. Then carbon dioxide is transported the opposite way, mainly as in the plasma as bicarb, then plain carbon dioxide and carbaminohemoglobin. You must remember oxyhemoglobin, deoxyhemoglobin. Carboxyhemoglobin is not normal. Carbaminohemoglobin is the one that's normal. Then we go to, so here's the control of breathing, your medulla and the pons. Of course, they're called chemoreceptors. Next, we go to chapter 24, digestive system. So this is an overview of the organs, 24.1. As you look at this one, you say, okay, I put my food here. What term are you supposed to remember? Ingestion. Then maybe you should remember mastication. Then deglutition. Right? Then here you say, okay, the substrate, polysaccharide, polypeptide, lipids. And then here the salivary glands secrete amylase that will break down the carbohydrates. So just trace the digestion of nutrients. You want to produce individual monosaccharides, glucose, individual amino acids, and individual fatty acids. How did you get to that? Okay. Peristalsis, segmentation, mass movements. The hormones, secretin, gastrin, cholecystokinin, amylase, lipase, and then the different proteases. Zymogen. The pH. Everywhere else is alkaline except in the stomach, which is acidic. The salivary gland is purely exocrine. The pancreas is mixed because it is exocrine and endocrine. Okay. The epithelium. So pharynx all the way to the upper 1/3 of the esophagus is stratified squamous, non-keratinized. Oh no, the whole esophagus. I mean the muscle. The muscle, the pharynx, all the way to the upper 1/3 of the esophagus is skeletal muscle. When you go down, from the lower 2/3 of the esophagus all the way down to the rectum, it switches to smooth muscle. Then when you get to the anus, it goes back to skeletal muscle, right? You have the external anus. So we did a lot of worksheets on the digestive system, so I'm really confident that you will be sure you take a look at the different features. So just looking at the pictures, this book, Tortora, has lots of pictures, that's why I like it. So remember rugae, chyme, bolus, chyme, feces. I don't think there were questions specifically the different cells, but you must know secretory cells, mucous cells, absorptive cells. I don't think we went into specifics for the parietal cells, the chief cells. You have to know the nervous regulation of the digestive system as well as the endocrine regulation of the digestive system. So while you're eating, you're tracing the flow, you say, okay, when the food goes to the stomach, gastrin is released to tell you to increase activity. Then when the stomach squeezes the chyme to the duodenum calls on the pancreas via secretin, and it also calls on the gallbladder via cholecystokinin. So remember that, okay? So here, see, maybe you can just take a look at this one, table 24.3. The pancreas, remember, is both exocrine and endocrine. The exocrine part is what we study for the digestive system, and they're called the acinar cells. So they're the ones that secrete all of these enzymes that is necessary to break down, right, to finalize the digestion. But remember that the duodenum has its own set of enzymes. The duodenum secretes enzymes that can break down the last bond. So you release the individual monomers. Now it can enter. So be sure you know that your amino acids and your glucose is able to enter the blood directly. But the fatty acids, they have to enter in the form of lipoprotein. So the fatty acids, they enter the columnar cells first as micelles. Then they enter the lymph as chylomicron. What's the function of the liver? Okay, portal circulation. What do the hepatocytes do once they receive all of this nutrient-rich blood? So just imagine they are grabbing all of these ingredients so that they can make your albumin, your bile, your vitamins. They can make all that. And so then when it leaves the liver, you now have your whatever you need, angiotensinogen, your everything, the proteins. They are also for detox. In the small intestine, villus, microvillus, segmentation. The parts, so like the small intestine has the duodenum, the jejunum, and the ileum, right? And then the colon has the cecum, then the ascending transverse. So be sure you remember the parts. What does mechanical digestion mean? What does chemical digestion mean? Right here, table 24.4. There's just a nice summary of everything. 24.5, the different enzymes. Don't forget, we said we cross out this lingual lipase, right? Cross it out. But cross it out. So the mouth is for digestion of carbs, the stomach for the digestion of proteins, and then the small intestine is for everything else. The appendix is not a digestive organ, it's a lymphatic organ. Then you poop, called defecation. And it's a reflex. Oh, right here, table 24.7. Summary of the organs of the digestive system and their functions. Then we go to chapter 25, metabolism and nutrition. So for this one, the point is you need ATP. And there's three sources: glucose, amino acids, or fats. So glucose is the first one we burn because it's broken down in the cytoplasm right away. And the first event for glucose is always glycolysis. And you know that glycolysis produces pyruvic acid right here. And if there's no oxygen, pyruvic becomes lactic acid, right? Creating acidic conditions, low pH. And we just discussed acid-base balance. Low pH is not good for the body. So in the presence of oxygen, you convert this to acetyl-CoA, send this Krebs cycle in the mitochondria, Krebs cycle, and electron transport chain. Then you get your then other breakdown products: ATP, water, what else? CO2 and heat. If you want to break the glycogenesis, glycogenolysis, you're familiar with that one. If you want to use your fat, the first step is beta oxidation, and then it's the same Krebs cycle, electron transport chain. But the side effect is there may be buildup of ketone bonds. If you want to use your amino acids, first step is deamination, which means there may be buildup of urea. But then, same step, acetyl-CoA, Krebs cycle, electron transport chain. So the lipoproteins, your HDL, LDL. So this is a nice summary. Nice summary of metabolism of lipids. Right here, table 25.16. Just a nice summary of everything already. You just have to add there your deamination, beta oxidation. So here, summary of metabolism, table 25.2. And the last part right here is thermoregulation. So you must know hyperthermia, hypothermia. By the way, there's an absorptive state and a post-absorptive state. So this metabolism, these events, are regulated by two main hormones, but all other hormones also affect this one. It's glucagon and insulin. So insulin is active when? Is it during the absorptive or post-absorptive? Then thermoregulation. Okay, then we go to chapter 26, the urinary system, which you just had your test. So I think you should be good on this one. Just had your, unless you have any, now you can ask. What was your question?
If you get your tubes tied or have them cut, why would you still menstruate? Because doesn't the is the fallopian tube like the connection of the ovary giving the hormones to your uterus?
Okay. Where are hormones released? That's what you should answer. Are they released into the fallopian tube? Where are they? Into the blood. Right? Into the circulation. Okay? So that's why you will not be menopause. Okay? Hormones are released into the blood. Okay. Any other question? That's a very good Confucius. When you do surgery, you do when they do tubal ligation, they don't cut the blood. They're very careful. They just cut they cut the structures of the fallopian tube, not the blood. If you sever the blood, there. Any more question? Any more question? Guys, one more example. Please try and study, okay? Study as groups or whatever. If you have been taking good notes, that will be your best friend. Take the time, just a little bit more, okay? Good luck. You don't have to show up tomorrow.